Okay, welcome back. You have made it through the toughest part of this chapter and probably the toughest part of this semester. So, round of applause for you for doing that. Okay, so let's talk about anaerobic. So we just talked about in the electron transport chain, at the end of it, that those electrons have to be picked up by oxygen, and that oxygen then will grab those hydrogens and form water. This is called aerobic respiration. That root word air, or, like air, like what's around you, the air. Anything that uses oxygen is called an aerobe. In chapter 6, we'll talk about some different types of aerobes. But some things are able to grow where there's not oxygen available. And they do something called anaerobic respiration. In anaerobic respiration, at the end of the electron transport chain, something that's not oxygen will pick up those electrons. Usually it's going to be some molecule with nitrogen or sulfur. It's not oxygen. That's what's important. Something that's not oxygen will pick up those electrons. So that electron transport chain keeps running even when oxygen's not there. Anaerobic respiration does not make quite as much energy as aerobic respiration does. Some parts of the Krebs cycle don't work quite as well. So we don't make quite as much ATP. So organisms that grow anaerobically, because they don't have quite as much ATP made, they grow a little bit more slowly. But they're still able to grow just fine without oxygen. An organism that doesn't require oxygen is called an anaerobe. They can grow without oxygen. What's interesting even is some organisms are killed by oxygen. So they are strictly anaerobic. They never do anything with oxygen. So if we summarize all of aerobic respiration, we have made 2 ATP by substrate level phosphorylation in glycolysis, 2 ATP by substrate level phosphorylation in the Krebs cycle, which made you 4 ATP. We made NADH in glycolysis, intermediate, and Krebs cycle that ended up making 30 ATP through the electron transport chain. And the FAD, we made 2 FAD, which gave us 4 ATP in the electron transport chain. So altogether, we made 38 ATP in a prokaryotic cell. Now, that is an ideal situation where every single hydrogen ion is going to come right back through ATP synthase and everything works perfectly. In real life, some hydrogen will diffuse away. In real life, molecules don't always react perfectly. So in real life, it's less than that. But in an ideal, perfect world, 38 ATP. And in a eukaryotic cell, that perfect world number would be 36 ATP. It's less ATP. Isn't that crazy? That's because it takes a little bit of ATP to move those carriers into the membrane, across the membrane, into the mitochondria. So we make a little bit less ATP in a eukaryotic cell, but still plenty enough. In anaerobic respiration, so some of these other molecules are our electron acceptors, some nitrogen or sulfur or something. You don't have to tell me this list. You need to be able to tell me in anaerobic respiration, the final electron acceptor is not oxygen. The ATP yield is not as high as aerobic respiration, and anaerobes grow slower than aerobes because with less ATP, there's less energy to grow. Okay, so you can tell me, just go back if you remember the electron transport chain, what's going on there. And you can compare the energy yield of aerobic and anaerobic respiration. Then let's talk about fermentation. So some cells, they can't do cellular respiration without oxygen, but they have some other way of surviving when oxygen is not there, and it's called fermentation. If you remember back a couple of videos ago, we talked about cells can either go fermentation or respiration. So here's the other option, fermentation. In fermentation, glucose is not totally broken down to carbon dioxide. It's going to be broken down to pyruvate, and then pyruvate can go a couple of different options, but it doesn't go all the way to carbon dioxide. Fermentation, the process itself doesn't make ATP. You are only surviving on those 1 or 2 ATP you made from glycolysis. That's all the ATP you're getting. What's important in fermentation is that it will keep NAD plus available to keep glycolysis running. That's all fermentation is doing, is keeping glycolysis running so your cell makes that little bit of ATP to survive. So it will not require oxygen, will not use the Krebs cycle or the electron transport chain. In fermentation, this pyruvic acid is going to be broken down to some fermentation end products. NADH is recycled back to NAD+. That NAD+ is back used for glycolysis. Because without fermentation, you will end up jamming up your electron transport chain. NADH won't be able to donate its electrons anymore. And you'll just have NADH building up until it backs up all the way to glycolysis. Fermentation prevents that from happening. So fermentation will take your NADH and turn it back to NAD+. That NAD+ keeps glycolysis running, so you keep making your ATP here. That's the whole purpose of fermentation. Without oxygen, you're still making a little bit of ATP. You are not going to have a ton of growth from this, but it's enough to keep your cells alive. Fermentation there's two different big types of fermentation that happen. One is alcohol fermentation. In alcohol fermentation, alcohol is produced. So it produces ethanol and CO2. So that pyruvate is decarboxylated, creates a CO2, and what's left is a 2-carbon molecule called ethanol. Alcohol fermentation is generally performed by yeast. So this is very intentionally performed by people who, want to make wine or beer or other alcoholic beverages that people for some reason enjoy sometimes. Lactic acid fermentation is when that pyruvate is going to be turned into lactic acid. And then there's 2 types of lactic acid fermentation. It could be homolactic fermentation that only makes lactic acid, or heterolactic fermentation that makes lactic acid and other stuff. What exactly it's going to be making in fermentation is going to depend on what kind of microbe you have, what kind of enzymes it has. And so because it's going to be different for different microbes, we can look at how a molecule does fermentation and use that to identify microbes. And in lab even, right, we do some carbohydrate fermentation, those little red tubes, to see if they're able to ferment. Does it produce carbon dioxide bubbles? We can use if it's able to do different types of fermentation to identify what kind of microbe there is. So this is just an example. Do not memorize this. Please don't waste time memorizing this. But you can see there's different end products created from fermentation. And you can say, oh, here I made lactic acid only. I know I have one of these types of bacteria. Here I made ethanol. I know I have one of these types of yeast. Here's whatever end products I've made. I can figure out what kind of bacteria or fungus I have. I can identify my organism using what kind of fermentation products it makes. Okay, a couple of other things that our cells can do. Besides breaking down sugar, we also can break down lipids. Lipids are a fabulous energy source for people. Not all microbes use them, though. For a lipid to be catabolized, first you're going to have to break lipids down to glycerol and fatty acids. It's broken down into these pieces by enzymes called lipase. Then each part can be metabolized on its own. Glycerol can come down and be broken down to go into glycolysis. Fatty acids can be broken down to acetyl-CoA to go into the Krebs cycle. Proteins are also a great form of energy. However, a big protein is too big to go through a plasma membrane, so it has to be broken down into its individual amino acids. And proteases and peptidases are those enzymes that are going to break that protein down into each little individual amino acid monomer. Those amino acids then can come through the membrane, but they're going to be converted before they can go into the Krebs cycle. They'll be deaminated. That means the amino group is removed. That amino group is turned into ammonia, and the rest of that organic acid will then enter into the Krebs cycle. Okay, so now you can tell me 4 compounds that can be made from pyruvic acid by an organism that uses fermentation, and what enzymes are involved in protein and lipid catabolism. Okay, see you back. We're almost done with this chapter. It's a big one, I know. See you back in our next video. Bye.